The webinar hosts are Dr. Anita Borges ma'am and Dr. Sumit Gujral sir. Moderators of today's session will be Dr. Dure Eden, who's Assistant Professor in HIMSA Delhi, and myself, Dr. Parul, Senior Resident in BPS GMC. A few reminders regarding connection interruptions, poll questions, and request for annotations. A few disclaimers. Disclaimers continued. Upcoming talks, that is on 3rd October, will be on similar topic, molecular diagnostic cases part 2 by Nikhil Patkar sir, Gaurav Chatterjee sir, and Shweta Rajpal ma'am. On following week, on 8th October, we'll have exam slides, histopathology and cytopathology by Dr. Neha Mittal ma'am. Let's start. Sir, I please request you to start sharing your screen.
Yeah, we'll do that. Yeah, it's visible, I guess, no? Full screen?
Yes, sir. It's visible. Full screen, yes.
So good evening, everyone. Me and Gaurav will take you through a few molecular techniques via few cases. So let's initially we'll see what is molecular biology. It is a branch of biology that deals with structure and function of macromolecules that include proteins and nucleic acid. These utilize nucleic acid like DNA, RNA, as well as proteins to understand molecular biology at molecular level. So in the molecular biology, we try to understand about human genome, which actually comprises of 22 autosome pair with 2 sex chromosome. And in these chromosomes, we have actually multiple genes that are there. We have 30,000 to 40,000 genes that are there, which have actually 3 billion base pairs that are there. So if we look at a cell that has a nucleus, it has these chromosomes have multiple genes as well as intergenic areas that are intergenic regions are there. In the genes also we have coding areas that are exons and non-coding areas that are in form of introns, and these whole sequence they are in form of nucleotides that are ATGC that are there. So if we look at the human genome, actually the most of the region is non-coding, and only 1% of the whole genome is actually coding genome, or that actually makes sense. So if we look at the central dogma of molecular biology, we have the starting material where we have DNA, which gets transcribed to RNA, a messenger RNA, which carries the message through translation and formation of protein in the cytoplasm. So this is a central dogma of molecular biology where DNA gets transcribed to RNA, RNA gets translated to a protein, which gives a functional meaning. So the central part of a molecular technique to any molecular technique is the polymerase chain reaction. Polymerase chain reaction was discovered by whom? So can Deba answer this question? So does anyone know? Yeah.
No.
Okay. Anyone else who knows who this person is? So Carey Mullis was a person who invented PCR, and he was awarded the Nobel Prize also in 1993. So Sayanta, can you tell me what happens in a PCR reaction? What are the components and what exactly is happening in a PCR reaction?
In a PCR reaction, we what we do, a specific sequence, we amplify it.
Correct.
First, through a primer, we anneal a 3' region. Then we amplify it, and then the cycle repeats. So
Okay.
we get many amplified sequences.
Okay, so in that, if we see what are the components that we will require for PCR, we would need a template, as you said, DNA will be required, right? Or RNA, whatever is there, that will be required. And then you need primers so that you can identify which areas to amplify. And then you need nucleotides that are there, and you need DNA polymerase that should help you for addition of these nucleotides. And you also need a buffer solution. So if you look at schematic diagram of what you see, so what happens, you have a double-stranded DNA structure, where primers are supposed to go and bind. Now, before primers have to bind, this double-stranded DNA has to break into a single-stranded structure. So basically, denaturation has to happen, where at a temperature more than 90 degree, at around 95 degree, your two DNA strand will get separated. Once they have got separated, a temperature of 60 degree around, this may vary according to primer. It is around 58 to 65 depending on your annealing temperature of your primer. It may vary, but approximately at 60 degree, your primers will go and bind to the site of interest. And then in the presence of DNA polymerase, extension will happen at a little higher temperature of 72 degrees Celsius, where your DNA polymerase will get activated and it will be adding the nucleotides in both directions. So what was initially at the end of one cycle of PCR, we will get 2 copies of DNA, double-stranded DNA, we will get 2 copies. So similarly, this is repeated for multiple cycles to get millions of copies of DNA. So basically, to visualize what is there, any change that is happening, what we are doing is we are amplifying a sequence. So this is called PCR, and this is one of the central technique of our molecular biology, where based on which our techniques are based. So now once we have amplified the product, how can we visualize these products? So according to the visualization process, our PCR technique may differ. So basically, there are various kinds of PCR. And so in today's example, I had given example of RT-PCR, reverse transcriptase polymerase chain reaction in a case. So let's go through that case and see what is there. So can Tapan read the history of this case?
So case 1, a 55-year female with dragging sensation in the abdomen. On examination, the spleen was palpable 10 centimeter below the left costal margin.
Correct. So what do you think till now? What possibility will you think with this history?
Ma'am, a dragging sensation of the abdomen with a splenomegaly, you will try to rule out the causes of individual splenomegaly.
Correct.
So we'll take further histories to rule out any causes that can be either hepatic, or there can be congestive splenomegaly, there can be hematological disorders which can cause splenomegaly.
Now we go to the peripheral smear where you are getting this kind of morphology. So what is your prominent finding here?
Ma'am, there is hyperleukocytosis with a myeloid predominance.
Correct.
Showing predominantly neutrophils as well as immature myeloid series, myelocyte, metamyelocytes. And we can also see few eosinophils, few dysplastic forms are also noted. Few blast-like cells are also seen. And
so you mean this would be blast-like cell? Right?
Yes, ma'am.
Shweta, can you tell him?
Yes, I'm going to laser pointer. Yes, sir.
Any basophil we can show.
Sir. Yes, sir. So this you mean is blast-like cell where with a prominent nucleoli, scant amount of cytoplasm. Here we are seeing this would be, I think, promyelo, because nucleoli is kind of visible. Then here we have myelocyte. These are eosinophil. This is another blast that we are having. And this should be a basophil. Here we can see coarse granule that is there. They are better basophil in bone marrow picture that we can go. So with these finding that we have leukocytosis, shift to left, basophilia. Here we cannot call myeloid preponderance. Why can't we say in peripheral blood myeloid preponderance word?
Because we cannot say whether the other series has been suppressed in the proliferating organs, and the RBC's picture as well as the appears normal in the smear while reduced also.
Correct. So actually in bone marrow, you are supposed to have normal erythroid precursors, but in peripheral blood, you are not supposed to have erythroid precursors, right? So we cannot call the use the term myeloid preponderance in peripheral blood finding. What we can say is that there is leukocytosis, there is shift to left, and there is basophilia. Now, what would you want to do next, Vishwabandhu?
Advice for bone marrow aspirate.
Bone marrow aspirate. So do you really need bone marrow aspirate? Like peripheral blood is there. Do you really need bone marrow aspirate in such cases?
I think we should need.
Sorry?
I think we should need to rule out.
Yes, rule out what?
The cause like CML.
Okay. See, for example, here I went ahead and performed FISH in this. Okay, so we have green signal that is coming from the BCR, and we have red signal that is coming from ABL. Okay, we have these yellow signal that are coming from BCR ABL fusion. Like we have two yellow signal here and one red, one green separate signal here. So this is BCR ABL fusion. Now I have got this BCR ABL fusion here. Then do I really need to go ahead for bone marrow? Because CML we have confirmed. And we had 2% blast suppose in the peripheral blood. So do we really need to do bone marrow?
We can classify the phase, whether it is a chronic phase or blast crisis phase.
Correct, correct.
Identifying the number of blasts. So
correct.
it can help in the prognosis.
Correct. So we have a diagnosis where we know there is reciprocal translocation of BCR-ABL has happened, where because of formation of BCR-ABL fusion, we will have constitutive activation of BCR-ABL gene, and that will lead to formation of chronic myeloid leukemia. So yet we went ahead and performed bone marrow because it will help us to distinguish between chronic phase and blast crisis, as well as cytogenetics is usually possible only in bone marrow because metaphase we get in bone marrow and we don't get that in peripheral blood. So apart from that, biopsy will also help us to look at fibrosis, and biopsy also may identify nests of blast. So here in the bone marrow aspirate, what is the finding here? Preeti, would you like to describe?
Yes, ma'am. The bone marrow aspirate of the particle, we are able to see it is a cellular particle.
So this age was 50 something. So cellular means, do you want to commit to hypercellular, hypocellular?
Hypercellular. It is a hypercellular aspirate.
Correct. Then can you see
increased,
these things? Yeah, increased.
Megakaryocytes.
Correct.
Megakaryocytes are also there. With increase in myeloid cells.
Correct. So now here we can use the term myeloid preponderance. And what is this cell here, Preeti?
It is a basophil.
Yes. Okay. So we have myelocyte, metamyelo, we have few eosinophil that are there. We have basophil. So shift to left. And so what is this cell?
Nucleated RBC.
Correct. So we have basophilia, we have very few nucleated RBC. Most of them are myeloid cells that we can see. And blasts were also less than 5% in marrow also. So we have a diagnosis where we have done bone marrow aspirate, where we are sticking to chronic myeloid leukemia chronic phase. And we have done FISH. Now, do we want to do anything else? Deba.
Ma'am, we can go for PCR.
Okay. So how will PCR help us now? Because in PCR also we'll come to know that it is BCR-ABL, right? How is PCR helping us?
Ma'am, we can go for quantitative PCR to see the quantity of the amount of BCR-ABL fusion is there.
So suppose I get amount of BCR-ABL as 100, or if I'm getting a 70 ISNCN, let's say that way. So how is it different? Like how is it making a difference for a diagnostic patient?
Ma'am, it can help in monitoring the therapy in future, not exactly for diagnosis.
Okay, okay. So basically for monitoring of therapy, if it will help, we just want to know what kind of BCR-ABL transcript is there. So at baseline, are we getting P210, P190, which transcript we are getting? That becomes important for PCR. Another thing that we also do is conventional karyotyping. So in conventional karyotyping, what is the finding here? Sayanta, do you want to tell?
Yeah, in this, the Q arm of 22nd chromosome is shortened, whereas the Q arm of 9th chromosome is of a larger length, and this denotes a translocation between 9 and 22 chromosome.
Okay, so for which of the chromosome do I call Philadelphia chromosome? Chromosome 9 or chromosome 22?
22.
Okay, so the shortened chromosome 22 is called Philadelphia chromosome, correct? So here we had translocation 922 that was there in the karyogram. So we went ahead and performed RT-PCR, and it is mandatory to identify the type of transcript here. So what if we did a quantitative kind of PCR at the diagnostic time point? This won't really help us, because in case it comes positive, well and good, but if it comes negative, it will not help us, because can be variant transcripts that may be happening. Like the common translocation of BCR-ABL happens between the exon 14 of BCR and exon 2 of ABL. But sometimes there may be exon 3 of ABL involved instead of exon 2. And atypical BCR breakpoint also may happen, where breakpoint in exon 6, exon 8, exon 19 may happen. So these cases will not be identified by your quantitative PCR. So in qualitative PCR, whereas you can do a multiplex PCR where you are amplifying a control area in the BCR gene. Here in the control area, you can see a product of 808 base pair. And in case you have E13A2, that is breakpoint in exon 13 of BCR and exon 2 of ABL gene, you will get a product of 310 base pair. E14A2, you will get a product of 385 base pair. And for your E1A2, that is P190 kind of product, that is your the one which we see commonly in BALL, you will get a product of 481 base pair. So in this same reaction, you are getting products of different base pair, and you will be able to identify what transcript is there. You can also go for variant PCR of BCR-ABL, where you can amplify the area of exon 3 also, and exon 6 and 8 of your BCR-ABL. So here what we are doing is we are using multiple primers, and whichever is present will amplify. So we can identify whichever transcript is there easily in case of RT-PCR at baseline. But in real-time PCR, what happens is, where you want to quantify, you will be able to amplify only one small area. So for real-time PCR, for the major transcript, that is E13A2 and E14A2, is a different type compared to what is there for E1A2, that is P190 transcript. So for example, a BALL case that comes for monitoring, for that the real-time PCR that we'll be putting will be different from compared to the case that has classical E14A2 transcript that will be coming for follow-up. So that is why we have to identify which transcript was present at baseline. Gaurav, do you want to add anything? Yeah. So in if we look at the BCR-ABL, the commonest transcript that we get is E14A2 in 63% cases, and E13A2 is there in 20% cases. But even in CML, we can get even A2 in 1% cases. So we have to identify what transcript was there in baseline so that exactly the same transcript we can identify in real-time PCR during monitoring time point. Here, if we look at this PCR, so Tapan, do you want to interpret? This is our patient. Okay, so we are getting two bands. One is at 808 base pair. First of all, on the right, we have molecular base pair. So this is 100 base pair. This is 200. This is 100, 200, 300, 400. It goes this way. And this is 1000 base pair ladder. So now compared to this, if we try to compare, so this is 100, 200, 300. So this band is near around 300 base pair, and this band is nearing around 400, and this band is nearing around 500. So similarly, this upper band is nearing around 800 base pair. So in our patient, we are getting two bands. One is at 800 around base pair, and other one is nearing around 400 base pair. So do you want to interpret, Tapan, what are we seeing in this case?
Ma'am, in our case, the patient's band is resembling the 385 base pair band, that is E14A2.
Correct.
Most likely on the transcript we are dealing with is the E14A2.
Okay. So in this patient, we are having E14A2. Now here, what is this band that we are seeing? That 800 base pair band, what is it? So if I go back to the example, here I had shown that we are also putting primer for the control region. That will give us a band of 800 around base pair. So this we will be seeing in every case. Okay. So now suppose this first case that is there, it is showing us only band of 808 base pair. So, but there's no other band that is there. So what will you consider this case as? Control.
Control.
Okay. So Preeti was saying something. So the control primer or control region has worked, but there's no other band. That means this case is negative for PCR label. Okay. But now at the same time, if I'm looking at this link, if I tell you that I had put a PCR for this also, but there's no band at all, can I call this negative?
When the reaction has then not occurred because the control band is not there.